we're going to start with the equations. You start with a glucose molecule, C6H12O6 plus 6O2, which is oxygen, that produces 6H2O, water, and 6CO2, carbon dioxide, and lots of energy. This energy is in the form of ATP. So ATP is made up of ADP, adenosine diphosphate, and another phosphate group. But these two things don't like to be together. So in order to put them together, you need lots of energy. And this is how the energy ATP is stored. So when you break it, all of a sudden you have a bunch of energy that you can use for something else.
The first step of cellular respiration is glycolysis. So within the cell, we've got the entire cytosol in the cellular space, and then certain organelles that have specialized functions. Glycolysis happens outside of the mitochondria in the cytosol area. In order for glycolysis to occur, you need one glucose molecule. No oxygen is required for glycolysis to occur. Now, glycolysis splits the glucose molecule into two pyruvate molecules that have more energy stored in them as well, but it also releases some energy in the form of 2 ATP. NADH also takes electrons from the glucose and brings them to the newer stages of cellular respiration. After glycolysis occurs, the pyruvate molecules have to be converted into something called acetyl-CoA for more energy to be released from them. This also means that these pyruvate molecules have to move from the cytosol outside the cell that I mentioned before into the mitochondrial matrix, which is essentially like the center of the mitochondria. There are 2 pyruvate molecules per glucose. This is just showing the breakdown of one pyruvate molecule into acetyl-CoA. Some CO2 is released, and coenzyme A enters in order to make acetyl-CoA. Electrons are also transferred and accepted by NADH.
Alright, the next step in cellular respiration is the citric acid cycle. So let's have acetyl-CoA. You're going to add that to this 4-carbon molecule, oxaloacetate, to create a 6-carbon molecule. Once this 6-carbon molecule is created, the CoA part of the molecule is going to leave. So we have acetyl-CoA, 6 carbons, CoA is gone, still have 6 carbons in a citrate molecule, which is where the citric acid cycle gets its name. Then we're going to do that thing again where we take NAD plus and we remove an electron from this compound right here to create NADH. So now we have an NADH produced that just took a pair of electrons, and then carbon dioxide leaves, also producing a 5-carbon molecule. We do the same step again where we convert NAD plus to NADH and get rid of carbon dioxide to make this a 4-carbon molecule. Then we go through and we use a molecule similar to ADP, GDP, and we produce GTP by removing a phosphate group. This FAD molecule is very similar to NAD plus and NADH. So we take the FAD, remove 2 electrons from our carbon molecule right here to produce FADH2. And then we're going to do the NAD plus thing again where we remove 2 electrons and produce NADH. And we're left with this oxaloacetate molecule that we had before.
Basically, the electron transport chain is a series of proteins along the mitochondrial inner membrane. And so first we'll start off with NADH and FADH2. They are our electron carriers, and they're passing their electrons to complex 1 and complex 2. And then from there, the electrons will move down these proteins. This action pumps the hydrogen protons into the intermembranous space from the matrix. Those hydrogen protons are basically stuck there until they reach the ATP synthase, which is this protein here. And then this protein basically acts like a rotor where each turn, ADP and a phosphate group will form to create ATP. And then the ATP synthase creates about 32 to 34 ATP within this process. The electrons that move down these proteins will basically form H2O because the electrons will be accepted by the oxygen, which is found in the matrix, and that oxygen with these hydrogen in the matrix also will form water. Anaerobic respiration is a way to make ATP without the presence of oxygen. So we have lactic acid fermentation and alcohol fermentation. Both are kind of like an expansion of glycolysis. And glycolysis itself can produce 2 ATP without the presence of oxygen. But we need fermentation to constantly make the regeneration of NAD+, which will end up being NADH with the addition of the electrons when glucose becomes 2-pyruvate. So in lactic acid, once the glucose becomes 2-pyruvate, the pyruvate will then convert into lactic acid with the addition of the 2 electrons from NADH. We also have alcohol fermentation. It basically works the same way as lactic acid fermentation with the constant regeneration of NAD+ for glycolysis, except this time when the glucose goes to pyruvate, the pyruvate will then become acetaldehyde, which will exactly release carbon dioxide during that process, and then the 2 acetaldehydes will become ethanol.
Question 1. What are the parts of cellular respiration? Question 2. Where does glycolysis occur? Question 3. Where does the citric acid cycle occur? Question 4. Where is the electron transport chain located? Question 5. What is needed for this process to occur? What happens if there is no oxygen? How much energy is produced? Why is oxygen used? How much water is formed? And finally, how is energy carried between the steps of cellular respiration?